Thank you for the opportunity to present our work here. Yeah, I'm kind of stepping in for Alessio here, who unfortunately couldn't make it. So I've got the pleasure of introducing you to some of the ways we use 19F NMR for drug design of E3 ligases and targeted protein degradation. I'm going to use the words targeted protein degradation and PROTACs a lot, so I'm going to spend one slide on actually introducing these concepts. So our lab is mostly interested in E3 ligases. Those are the enzymes that regulate the degradation of proteins for the proteasome. And specifically, we're interested in these CRLs, choline ring ligases, which are multi-protein component complexes that have these modular substrate receptors that recognize the target proteins and form complexes with those where then the ring domain of the IBX protein can assemble ubiquitin chains on the target proteins, which leads to polyubiquitination of the protein, which is a degradation signal that is recognized by the proteasome, and the protein is then degraded. A lot of these free ligases are interesting drug targets, but they are traditionally considered undruggable since these ring domains don't really have a classic active center that you could easily target. However, you can target, at least to some extent, the substrate receptors by protein-protein inhibitors, protein-protein interaction inhibitors. This is an example for one of those that we've developed, and this blocks the first step where the substrate is recognized by the CRL, and then inhibits the rest of the cascade. However, if you have such an inhibitor, you can do something fairly cool with that, and that is you can attach a linker to this and use this linker to attach a ligand for another protein that you want to get rid of. This is a molecule we call a PROTAC, a proteolysis targeting chimera. And basically what that does is that it forms the formation of a ternary complex between the CRL and the protein of interest, which then, all going well, leads to polyubiquitination and degradation of this protein. The major limitation in this approach is that there are very few E3 ligases for which we have good drug-like ligands. One example for that is VHL, which is this substrate receptor for choline 2. And this naturally binds the transcription factor HIF1 after phosphorylation of a tyrosine. And starting from, hydroxylation of a proline. Starting from hydroxyproline, we've developed high affinity binders for that with about less than 200 nanomolar affinity. And we have used those as the basics for PROTACs for a number of proteins successfully. And also other people have designed a lot of PROTACs using that. More recently, the team I've been working on has been trying to expand this, and we've developed ligands for a new E3 ligase, which is SOX2, suppressor of cytokine signaling 2, which recognizes a phosphatyrosine and cytokine receptor intracellular domain, and is otherwise a relatively similar protein to VHL, as you can see from the similar color code. It uses the same adapter proteins to interface with choline 5. And I will tell you two aspects where we use 19F NMR in these for these systems. So from how I explained to you how PROTACs work, I might have to explain briefly why we need biophysics in these for these systems. Since it sounds a bit like we have two independent binders for two proteins, and as long as these are well characterized, we should be able to predict the complex formation. But that's not actually that simple. If we look at the crystal structure of one of these induced complexes between a neosubstrate and VHL, you can see that they're quite large, that they have quite large protein-protein interfaces induced by the PROTAC, and these can favorably contribute to complex formations. And we often observe strongly cooperative binding, where the ternary complex has about 30 times higher, up to 30 times higher affinities than just the binary binding. We could sometimes also have clashes that lead to reduced binding. So in order to successfully design PROTACs, we need a lot of biophysics on these complexes, and we really use the whole toolkit. There's a lot of SPR and FP. Of relevance for this audience, we think there's a lot of potential to use integrative approaches, and a lot of these protein-protein interactions are quite transient and proximity induced. And we think that protein-observed NMR, SACs, and structural mass spec have great potential for that. We recently wrote an opinion piece on that, thinly disguised as a review, if you're interested in that. But what I'm gonna focus on today is our work on 19F NMR. Being basically the last speaker in this session on small molecules, most of the concepts have already been explained. Nevertheless, I'm gonna repeat some of that. 19F NMR is, of course, a very nice nuclei to work with. It is very sensitive, since it's the only natural fluorine isotope, and it has quite a high gyromagnetic ratio. And it's also mostly absent from biological samples, so we have very little interference from other components in our sample. And also medicinal chemists love to put fluorines into their molecules, which I very strongly encourage them to do as well. So we usually have at least one or two signals that we can look at in our compounds. And also most labs are already equipped to do at least some fluorine NMR, since you can usually just tune the proton channel of your probes to detect fluorine NMR if you don't have a dedicated fluorine probe. Fluorine NMR is a bit different from protein NMR in some ways, and that's because fluorine is the most electronegative element, and that leads to very strongly polarized bonds, which leads to a few properties that are that we probably don't know that way from protons, like very strong J coupling. You can often observe up to 5J couplings. There's a very large spectral window, and you've got a very strong sensitivity of the chemical shifts to their chemical environment. And also these strongly polarized bonds lead to very strong chemical shift anisotropy, which means that unlike from protons, where we know that usually we can improve our resolution by going to higher fields, that typically doesn't work with fluorine NMR. So you typically get the best resolution at relatively low fields. But it means that fluorine is very sensitive to changes in the to relaxation as well. So CPMGs work very well. And we've already heard a lot about direct screening for fragments with 19F NMR. I'm not going to talk about that. Our lab doesn't really have much experience with that, but the advantages of that are quite evident. What we use a lot is the already mentioned before displacement assays using 19F spy probes, where we have a spy molecule that binds to our protein of interest, usually D3 ligase, and we measure the competition with a competitor to that. The nice thing about these assays is that they're quite quantitative, and we can actually get affinities for that. The difficulty is usually to find a good spy molecule. Traditionally, a lot of those are either derived from natural substrates of the protein or are early stage hits from fragment binding, which then usually have relatively low affinity. So a PhD student in our lab, Guillaume, spent some time optimizing these assays a few years ago, actually, for VHL, and he wasn't content with just using a spy probe that came from fragment binding. He wanted to design one from the ground up to be to have the optimal properties. And what that really means is that we want to have a large window between the CPMG signal for the protein-bound spy and the free spy. Physically, that translates into a large difference in the spin-spin relaxation rate, so a large C2 contrast. GUI designed a number of possible spy molecules by varying the position of a CF3 group in a scaffold for VHL ligand based on hydroxyproline that we already know we can get to very high affinities, and that we know how to fine-tune the affinities by changing a few other substituents. When he when he plotted the C2 contrast he observed for these molecules against the affinities that we see. You get a bit this bell curve-like shape that becomes a bit more obvious when you look at C2 within one series, where not unexpectedly, very strong binders do not give high contours. That's just because those have relatively slow exchange rates, and once bound, stay bound for the duration of the experiments. Only a relatively relatively small proportion of the molecules actually engages the protein, and for very weak binders, we also observe very small contrasts, since these barely engage the protein. However, even in the range, in a more reasonable affinity range, in the hundreds of micromolar range, or in the tens to hundreds of micromolar range, contrast can differ very strongly between different binders, and so that really shows that there is some potential to fine tune the contrast ratios that you can obtain using these, and to optimize your binders. Once we settled on a good SPI molecule, we benchmark those against SPR assays. So those are the results from single CPMJ delay, single protein concentration titrations with a competitor, where we changed the concentration of the competitor, and we got a very good correlation to SPR assays. These assays work especially well for very weak binders, for example, from fragment screening, as long as they bind to the same pocket. And I think there is some potential for those, especially in chemistry labs that might not have access to an SPR instrument, to use them alternatively to that. When these assays were developed, we were already the work on VHL ligands had already been progressed quite a lot, so we already had quite high affinity binders, and especially we were already working on PROTACs derived from these ligands. So we were interested in seeing if we could use those assays also to characterize our PROTAC ternary complexes. So the main factor we're interested in here is cooperativity. So do we form favorable protein-protein interactions between the two proteins? And the factor the number we usually use in the field to describe that is this alpha value, which is just the binary dissociation, or in this case, inhibition coefficient between the E3 ligase and the SPI and the ternary, between E3 ligase and SPI in the presence of the third interaction partner. When GUI tried this with a series of PROTACs for these PROTACs that show negative cooperativity, so that there's weaker binding in the ternary case than in the binary case, he could very well reproduce the values that were obtained in the literature. But when he looked at one of our most successful PROTAC, and a very strongly cooperative PROTAC that has about an alpha about 30 measured from SPR, he wildly underestimated that number. And that is probably because we're really running into the tight binding limits for these assays, so they really can't detect reliably quantify KDs that are lower than about half the protein concentration, and that translates to about 500 nanomolar in these assays. So for very strong binders, these assays are quite limited. They do, however, work quite well for weaker binders. For example, this fluorinated version of MZ1 that has about 10 times reduced binding, we can reliably observe cooperativity for those. One caveat with these assays when you use them for ternary complexes is also that you need to make sure that your SPI molecule does not interact with the third binding partner. In our case, the third binding partner is BRD4, which is a very, very ligandable protein. And so we had a few SPI molecules that had very favorable properties for binding to VHL, but unfortunately they also showed some binding to BRD4 and were not useful for ternary complex assays. So that is something you need to check for. Yeah, with that, I'm going to switch to the second part of the talk that I want to talk where I want to talk about the work on suppressor of cytokine 2 signaling. This is a substrate receptor for Calin-5 that recognizes an epitope in the intracellular domain of cytokine receptors after they get phosphorylated at a tyrosine. And that provides a negative feedback loop for cytokine signaling to avoid things like cytokine storms that you sometimes see in infectious diseases. Yeah, our lab has had a very long-standing project to target these proteins. That is mostly work from Nikolai and Saraf, but all in all, it has been 3 generations of postdocs working on that. And the starting point for that has been phosphotyrosine, which for being such a small fragment has quite a decent affinity to the protein with about 200 micromolar. And through a series of structure-based drug design, we have progressed that until we ended up with binders of about 200 nanomolar affinity. This has been about 50 crystal structures with different ligands solved in between there. And we've characterized all these ligands extensively using SPR and LTC and also 19F NMR. And at this stage, we realized that we were growing the fragment. Well, we accidentally realized that we were growing this fragment towards a cysteine in the binding site that we could target covalently, and so that gave rise to this molecule, which is MN551, which has this chloroacetamide warhead that covalently engages our cysteine, and yeah, is a very potent inhibitor for the SH2 domain of SOX2 in vitro. If we look at this molecule, it's really the kind of molecule that makes a medicinal chemist cry. It has this this chloroacetamide, which can be quite notorious in terms of off targets and is not very stable. The backbone is essentially a peptide backbone, so it clearly is still peptide derived, and worst of all, it still has this phosphate group that is really important for binding to SOX2. And the problem of the phosphate group is that it's strongly negatively charged, and it really limits the cell permeability of these compounds. So to get around that, Nikolai tried to design some PROTACs that would mask the negative charge of the phosphate group. Those basically fall into 2 categories. There are these PROTIDes that come from the nucleotide analog field, and they're basically arryl based, and these ester-based POM protection groups. And when Saraf tested those in cells using SETSAR assays, where we measure stabilization of the protein in cell upon treatment with the compound, we realized that really only the POM protected PROTAC increased cellular engagement significantly over the control with the unprotected group, whereas the others that either had no significant change or made things significantly worse. And we were a bit puzzled by that. We couldn't really explain these differences. So at that stage, I had the somewhat stupid idea to try to use NMR to follow the fate of these molecules in cells. So we decided to use 19F NMR for that. And basically, it's a relatively simple experiment. We had a dense cell suspension. We used K562 cell line. That's a human leukemia cell line. For that, that is quite tolerant to being handled in suspension. So we had a 70% by volume cell suspension approximately, treated them with the cells, transferred into a Shiigemi tube, and then measured a series of 19F NMR spectra. If we're looking back at this molecule, there is this fluorine that is quite distant to the phosphate group, So originally we didn't really expect that would be sensitive to the phosphate, and we tried introducing additional fluorines. That didn't go very well, but it turned out that the fluorine that we already had in there was actually sensitive enough and showed a small chemical shift perturbation upon the protection of the phosphate. So here is the data that we obtained for for MN714, so the POM protected PROTAC that actually worked quite nicely. And immediately after addition of the PROTAC, we see one big peak that we think corresponds to 714 with that has a bit of a shoulder, and over time this shoulder grows and becomes the dominant peak, whereas the originally main peak seems to disappear. And we believe that is due to unmasking. So to confirm that, we lyse these cells. They're mammalian cells, so they're very easy to lyse just by freeze thawing. Yeah, we still observed a single peak. And when we spiked the sample with the free MN551, these peaks overlap very well, so indicating that we're indeed forming MN551. Just to confirm that, we spiked with MN714, which is very badly soluble under these conditions. It's not very highly soluble in media. We observed a difference in chemical shift that fitted very well to what we saw in cells. So we're quite confident that we're actually seeing the unmasking. Yeah, so MN551 is formed in cell, and it enriches to hundreds of micromolar in the cells. And subsequently, using proteomics, we can also show that it actually engages the cysteine we want to target, which makes this the first covalent cell permeable inhibitor on SH2 domain, which are, as a domain, is traditionally considered undruggable. That leaves the question of why our PROTACs don't work. And that seems to be really a combination of different factors. So when we repeated this experiment, you can see the time scale is now considerably longer. We're looking at about 6 hours here. And initially, we observed very weak signal. So the compound is very badly soluble in the media. And only after about 2 hours, we start to get a decent signal from the cell suspension. So it does seem to take a long time to get into the cells. It's badly soluble. It has very slow cell permeability. And even though we see some unmasking, it never really reaches, it never goes to completion. So even after 6 hours, we only observe about half of the compound being unmasked. So there are really several limiting factors for these PROTAC drugs. With that, I'm coming to my very brief summary. So I hope I could convince you that 19F displacement assays are a really great way of quantifying, especially weak interactors. They do offer an interesting alternative for especially chemistry labs that might not have access to an SBR instrument, and probably will have access to an NMR of some description. I would also like to really make the point that 19F NMR is not just limited to in vitro applications, it can be really applied to cells and other complex mixtures, and is very versatile in that regard. I would like to thank my supervisor Alessio Ciulli for supervision. The work I've presented here on the displacement assays is mostly done by Guillaume, who's a PhD student, was a PhD student in the lab, and left a few years ago. This is the team that did the work on SOX2. Most of the ligand design was done by Saraf and Nikolai. More recently, Dylan and I have been taking over this project. And we've been supported by the NMR facility in Dundee, originally by Dan Fletcher, and more recently by Olga Semenova. Thank you very much for your attention, and thank you very much for the opportunity to present this work here.